Ladies and gentlemen, we are welcome to our YouTube channel. Let us appreciate our protein chemistry. So in this case, we are going to look at the sickle cell anemia. What is a sickle cell anemia? It's an inherited form of anemia, a condition in which there are not enough healthy red blood cells to carry adequate oxygen throughout your body. So, a sickle cell anemia is a inherited disease, it's a genetic disease. So, usually the disease is acquired through the genetic inheritance of either from a sickle cell parent or a sickle cell carriers, or from a sickle cell parent carriers. So, generally, in a sickle cell anemia, the red blood cells is not healthy enough to carry adequate oxygen throughout your body. And of course, like for example, a normal, a normal, a normal red blood cell is responsible for carrying four oxygen molecules. But in the case of a sickle cell, red blood cells only carry only one oxygen. So normally your red blood cells are flexible and round, moving moving easily through your body blood vessels. That is the normal red blood cell. It is flexible, round, and it can easily move through the red blood cell. So let's see what happened in the case of a sickle cell condition. So of course, in sickle cell anemia, the red blood cells become rigid and sticky and are shaped like a sickles or crescent moons. So in a sickle cell, the red blood cells become very rigid, sticky, and are shaped like a sickles or crescent moons. Like for example, I think you can clearly see here. This is the typical normal red blood cell. This is the normal red blood cell. Look at it here. It is the normal red blood cells here. But you see that in the case of sickle cell, you see that it becomes sticky sickle cell. Sticky. Look at it here. It becomes sticky sickle cells, and of course it is shape change like a crescent moons. And these irregular shaped cells can easily stocked in the blood vessels or in the small blood vessels, which can slow or block blood flow and oxygen to the fat of the body. So as a result of that, this stock or sticky form red blood cells will actually block or it will stock the blood vessels. So therefore, there will be a slow or blocked blood flow. And of course, when there is a blocked blood flow, it means that there will be low oxygen delivery. And of course, look at the normal hemoglobin here, the normal hemoglobin here, and then of course, this is the abnormal hemoglobin. What is hemoglobin? Hemoglobin is actually a protein that is found inside the red blood cells, which is responsible for the transport of oxygen. So that is for the red blood cells. That is for the sickle cell red blood cell. And then, of course, so what is the definition of the sickle cell? So sickle cell disease is a genetic disorder that affects erythrocyte, causing them to become sickle or crescent shaped. So it is a genetic disorder we usually inherit or child get the disease either through infected person or through infected parent or carrier parent. So generally the shape of the blood cells becomes sickle or something like crescent shaped moon. So the effect of these conditions are actually due to abnormality of the hemoglobin molecules that is found in the red blood cells. So the abnormality in the red blood cells is what actually causes the sickle cell disease, the abnormality in the hemoglobin, the abnormality in the hemoglobin molecules that is found in the red blood cells is what actually affects the condition. So that is the very important thing. So the sickle cell disease is due to the abnormality of the red blood cell. So then, the next thing that we are going to look at is so these are some basic features of the erythrocyte, which is red blood cells. So it is called red blood cells, and it produced through a process called erythropoiesis. Erythropoiesis is a process in which red blood cells are produced using erythropoietin. And of course, we have in the red blood cell, that is erythrocyte, it contains hemoglobin. It contains hemoglobin, which is actually the major protein that is found in the red blood cells that is responsible for binding the oxygen. And of course, it transport oxygen, and that is the major functions of the red blood cells. And the lifespan of the red blood cells is about 100 to 120 days. But in the case of sickle cell red blood cells, the lifespan significantly reduced to about 10 to 20 days. So those people with the sickle, the red blood cells have a lifespan of about 10 to 20 days. And of course, the red blood cells, it doesn't have nucleus, and of course, as a result of that, it depends solely on the glycolysis for energy. So that is why in many cases, whenever the glycolysis is having any problem or when there is any enzyme deficiency in the red blood cells, it easily causes anemia. Then the erythrocyte usually squeeze through capillaries, transport oxygen, and it is very smooth around, which is sticky and rigid in the case of the sickle cell patient. Then what is hemoglobin? Hemoglobin is the oxygen carrying pigment and is predominant protein in the red blood cells. So hemoglobin forms an unstable reversible bond with oxygen. So generally the bonding that occur between oxygen and the hemoglobin is unstable, reversible, because it needs to be reversible because hemoglobin is carrying oxygen from the lungs, transport them to the body tissue, and then deposit them there, recycle itself, and come back again to take another oxygen. So that is why it must be unstable and reversible. If it is stable and irreversible, it means that each oxygen will be actually transported with a separate hemoglobin, which is not possible. So it's like a car that carry a passenger, drop them, shuttle back, take another passenger and deliver them. So that is what happens. So the bonding should be unstable and reversible so that the oxygen can be easily dissociated with the hemoglobin when delivered to the expected body tissue and then come back. So when hemoglobin binds with oxygen, it is called oxyhemoglobin, and we call it oxygenated blood. And of course, the color is bright red. Then of course, when the hemoglobin dissociates with oxygen, that is delivered oxygen, so it's now become reduced. So we call it reduced hemoglobin or deoxygenated hemoglobin, and the color is usually purple blue. It's usually purple blue. So then, of course, each hemoglobin molecule is made of a four heme surrounded by a globin. So there are four heme and it is surrounded by a globin. And of course, the globin consists of two linked pairs. So in the red blood cells, sorry, in the hemoglobin, we have a heme component and we have globin. So globin is a protein component. Globin Globin is a protein component, is a protein component of the red blood cell. And of course, the heme is an inorganic component. It's an inorganic component of hemoglobin. And of course, in this heme is made up of, it is highly rich in an element ion, Fe ion. So of course, now let's look at the protein component of the hemoglobin. The protein component, that is the globin, is made up of a protein component. So this protein component is made up of two beta chain. It's made up of two beta chain and of course two alpha chain. Made up of two alpha chain and two beta chain. So now of course, the alpha chain, as we said, we have two alpha. We have two alpha, and each of these alpha chain is contain 141 amino acids. And of course, then for the beta, we also have two beta, which contain 146 and then 146 amino acids. So it means that in the hemoglobin, we have, as I said, we have globin, which is the protein component, and we have the non-protein component. So the protein component is made up of two alpha beta and two alpha, two alpha and two beta. So alpha, each of the alpha contain 141 amino acid, and each of the beta also contain 146 amino acids. So then let's look at, so we have different types of hemoglobin, and we have the normal hemoglobins. We have hemoglobin A, we have hemoglobin A2, and we have fetal hemoglobin, that's hemoglobin F. And then we have hemoglobin S, hemoglobin C, and hemoglobin E, which is the abnormal types of hemoglobin. So now let's look at the genetic basis of the sickle cell disease. So actually the change in the structure arises from a change in the structure of the hemoglobin. So we said that the shape of the red blood cells usually change to sickle shape, and this change of the red blood cells is due to the change in the structure of the hemoglobin. And surprisingly, just a single change in one amino acid is what actually causes hemoglobin to aggregate, and of course leading to a disease condition called sickle cell anemia. So in the of course beta chain, the beta chain of hemoglobin, because of course hemoglobin is a protein, so there is a specific gene in the DNA. There is a specific gene in the DNA that is responsible for producing the hemoglobin. So for the beta change, because when you said in the DNA, if you said in the DNA, it means that definitely we know that the DNA is present in the chromosome. So now the question is, the beta globin gene, usually the problem is emanating or is usually coming from the beta globin. That is the beta chain of the hemoglobin. So now the question is, what is the address? What is the address of the beta globin? So it is located at chromosome 11. So is it at the short arm, or is it at the short arm or long arm? P arm of the chromosome is the short arm, and the Q arm is the long arm. So actually it is located at the P arm, that is the short arm of the chromosome, locus 15. when we said locus is a point where the chromosomes are found. So where the genes are found. So beta globin, it is at chromosome 11, short arm of the chromosomes, locus 15. So you see, this is actually the normal, let's say this is a partial DNA sequence of beta globin. So you see that in this beta globin DNA sequence, we have CCT, GA, GC, GA, G also. And of course, you see that let's look at uh, the amino acids. So you see this DNA will be converted to produce a partial RNA sequence with with CCU, GAG, then GAG again. So this CCA or this CCU code for amino acid, it code for amino acids, it code for amino acid choline. And then of course the GAG code for amino acid glutamic acid. And then the GAG code for another glutamic acid. So this is actually a normal hemoglobin where we have a glutamic acid here. But in the case of the sickle cell patient, there is a mutation. We call it missense mutation. You see here is A, but here is T. So it means that A is replaced with T. So of course, the A will also change. So the pairing sequence, because A is changed with T, so it paired with A. So of course, this mutation of A replacing, or this replacement of adenine, which is A, with thymine, will now code for uracil instead of adenine. So as a result of that, instead for glutamic acid to be produced, then valine is now generated. So as a result of that, glutamic acid is a polar acidic amino acids. And of course, it's due to this polar glutamic acid that make the red blood cells to have its shape. But of course, when the adenine or when the thymine is replaced with uracil, we have G use G instead of GAG, which code for valine. And valine is a non-polar amino acid, non-polar amino acid. So as a result of this, the shape of the red blood cells change to sickle. And that's what leads to a condition called sickle cell anemia. It's just a replacement of glutamic acid with valine due to missense mutation in the partial sequence or in the sequence of the hemoglobin gene. And that's what leads to sickle cell disease. So, so this is the normal hemoglobin A, and then this is a sickle cell hemoglobin. Look at it. A sickle cell hemoglobin, they are not the same in terms of the structure. And all this is because of the replacement of glutamic acid with valine. And then so now let's look at the inheritance of the sickle cell disease. So if one parent has sickle cell traits, that is hemoglobin AS, it's a carrier. So the other genes does not carry the sickle cell hemoglobin at all. That is hemoglobin AA. So if you have hemoglobin AS, it means that you are a carrier. And if you are hemoglobin AA, it means that you have a normal hemoglobin and you don't carry the disease. Then none of the children will have a sickle cell. If you have one parent have hemoglobin AS and the other parent have hemoglobin AA. So you see here, there is hemoglobin AA. We have yeah, there are two parent, and of course, this is AA and this is AS. So there will be none of the parent will have a sickler. Sorry, none of the child will have a sickler. But of course, 50% of the child will be a carrier, and then 50% of the child will be normal. So you see, if you cross this A with this A, you have AA. This A with this S, you have AS. This A with this A, you have AA. And A with this, you have AS. So AS here is a carrier, and this one is also a carrier. So it carry the trait. But these are the normal because they have AA. So there is 1 in 2, that is 50% chance that any given child will get a one copy of the hemoglobin AS gene and therefore have the sickle cell traits. So it is equally likely that any given child will get hemoglobin AA genes and then be completely unaffected. So that is what happened in the case of one parent having uh, the trait, that is hemoglobin AS and then hemoglobin AA. So now let's look at a situation where we have, uh, for example, if both the parent have sickle cell, that is, they have the trait, but the disease doesn't here. So they have all hemoglobin AS and AS. So hemoglobin AS and AS, if you cross AS and AS, then at the end, you are going to have so there is a likelihood that after, if you have AS and AS, there is a likelihood that 25%, there is a 25% chance of giving birth of a child with anemia. So you see, after crossing, you will have one normal child with AA, two carriers, and then one anemic, one with the sickle cell anemia. So you see, it is 25%. You see, you have one S, so we have 1, 2, 3, 4. So 4 times 100 is going to give us 25%. So 25% of the child will likely come up with the sickle cell anemia. And there is also a chance in full chance, or there is a one chance in full child that one will be completely unaffected, and there is a chance, or there is a 1 in 2 50% chances that any given child will be or will get the sickle cell traits. So you see 50% here, AS and AS, they have the traits. They have the traits. So that is for this case. And then, so what happened if you are married to, if you have AS and SS? So there is a chances that 50% of the child will be and will have the anemia, and then 50% of the child will be a carrier. So no children will be completely unaffected. So that is why it is very, very important to do genotype tests to know which types of the hemoglobin you have, so that to avoid giving birth to a child with sickle cell disease is very, very important. So what of if you normal hemoglobin with you have normal hemoglobin AA, and then you are married with a sickle cell patient? So it means that all the child will carry the traits, but none will have anemia. None of them will have the sickle, but they are all going to be a carrier. So that is why there is a need for the genetic counseling before you get married. So then mechanisms. So when sickle cell hemoglobin gives off its oxygen to the tissues, so hemoglobin sticks together, which actually lead to forms a long rod form inside the red blood cell. And the red blood cells will become very rigid, inflexible, and sickle shaped, which is unable to squeeze through small blood vessels, instead blocks small blood vessels. And as a result of that, there will be less oxygen delivery to the body system. And then, so you see, due to the change in the shape of the hemoglobin as a result of the replacement of glutamic acid with valine, the red blood cells will form a long rod form. Oh, the hemoglobin inside the red blood cells form a long rods, and the red blood cells, as a result of that, the red blood cells form very rigid, inflexible, and sickle shaped, and will be unable to squeeze through the small vessels, which also leads to the less oxygen delivery to the body tissue. And of course, the red blood cells containing abnormal hemoglobin have a shorter lifespan, and normally the lifespan of the sickle cell patient is just 20 days. It's a 20 days instead of 100 to 120 days. And it is actually a chronic stage of anemia. So this is it. You see in the normal red blood cells, you see that the red blood cells can be able to squeeze and then deliver oxygen. So it can easily cross section of the red blood cells. So we have hemoglobin, look at normal hemoglobin. But in the case of the sickle shape, look at how it formed these key sickle cells. And of course, we can see clearly the red blood cells is unable to squeeze and pass through the small blood vessels. So what are the symptoms of anemia? So the symptoms actually vary from person to person and also change over time. So it include anemia. So sickle cell break apart easily and die, so leaving you without enough red blood cells, because it just only take 10 to 20 days instead of 120 days. So as a result of that, there is a likelihood that you will not be having enough red blood cells. And of course, if there is no enough red blood cells, it means that your body can get oxygen it needs to feel energized. So therefore, this will lead person fatigue, because remember that the most important thing in the oxygen system is that electron transport chain and citric acid cycle, beta oxidation are the major source of energy in the body. So therefore, if there is low oxygen delivery, it means that there is a likelihood that you will not get enough energy in your body. So as a result of that, you will not be energized and your body will be fatigue. And that's one of the even major reasons that cause the crisis of sickle cell anemia, because there is no production of enough energy. So that will lead to fatigue. So then another symptoms, the episode of pain, periodic episode of pain, which is called crisis, are a major symptoms of sickle cell anemia. So pain develops when sickle cell red blood cells block blood flow through tiny blood vessels to your chest, abdomen, and joint. So as a result of that, pain can also occur in your bones, because the red blood cells is blocked, so no oxygen delivery. So there will be chest pain, abdomen, and joint, and also bones pain. So the pain varies in intensity and can last for a few hours to a few weeks. Some people have only a few pain episodes. Others have a dozen or more crises a year. If a crisis is severe enough, you might need to be hospitalized Some adolescents and adults with sickle cell anemia also have chronic pain, which can result from bone and joint damage, ulcers, and other causes. So you see, because of that low oxygen delivery, red blood cells are blocked. Oxygen are not delivered to the chest, to the abdomen and joint, and of course, there will be crisis because there is no energy there. And that is why a lot of this crisis occur. And then also painful swelling of hand and feet. So the swelling is caused by the sickle shaft and the red blood cells blocking blood flow to the hands and feet. And that's what causes the swelling of hands and the feet. And then there's a frequent infection. Sickle cells can damage an organ. We know that we have an organs that usually fight infections, which is spleen. So usually the skill cells can damage spleen. So if the damage spleen, what happens? It means that the infections can easily get into your body, and that is why sometimes it is always advisable for the patients having sickle to be taking antibiotics because the crisis damage the spleen, which lower the immunity of the body. So then delay growth. Red blood cells provide your body with oxygen and nutrients so you need for that are needed for the growth. So a shortage of the healthy red blood cell can slow growth in infant and children and delay puberty in teenagers. And of course, all this is because the nutrients and the oxygen that delivered by the red blood cells is not enough. There is also a vision problem. So tiny blood vessels that supply your eyes might also become blocked and sickle cells. So this damage the retina, so the portion of the eye that processes visual images leading to vision problem. And then the complications. So sickle cell anemia can lead to a host of complications, including stroke, because you need oxygen. So the sickle cell blocks blood flow to the area of your brain, and there is also low oxygen delivery in the blood. So signs of the stroke include seizures, weaknesses, or numbness of your arms and legs, sudden speech difficulties, and loss of consciousness. So if your baby or child has any of these signs and symptoms, seek medical treatment immediately. A stroke can be fatal. And there is also acute chest syndrome. This is a life-threatening complication that causes chest pain, fever, and difficulty breathing. And of course, acute chest and syndromes can cause by a lung infection. And there is also pulmonary hypertension. So people with sickle cell anemia can develop high blood pressure in their lungs. So, and also cause organ damage, blindness, and leg ulcers. It also causes gallstone. And of course, the breaking down of a red blood cell produces a substance called bilirubin. So a high level of bilirubin in your body can lead to gallstone. So then diagnosis. So the diagnosis of the sickle cell disease, a blood test can check for the hemoglobin S. That is a defective form of the hemoglobin that underlies sickle cell anemia. In the United States, this blood test is part of the routine newborn screening done at the hospital, but older children and adults can be tested too. So you can get the blood cells and test for the presence of hemoglobin S. And of course, in adult, a blood sample is drawn from a vein in the arm. So in the young children and babies, the blood sample is usually collected from a finger or hair. So if the screening test is negative, it means that there is no sickle cell gene present. But if the screening test is positive, then further tests will be done to determine whether one or two sickle cell genes are present. And of course, the check for a low red blood cells, that is anemia, will also be done to also confirm the count for the red blood cells. And then there is also counseling. Genetic counseling. So usually, if two tests can be used to have expected parent to find out if the child is affected. So that is why it is always important, even if you are married and you have uh, AS and AS, you know that you are AS and AS, so you may actually need to know the status of your child. So, of course, amniocentesis synthesis usually is done when the pregnancy reach 14 to 16 weeks. So the sample of amniotic fluid in the womb for the genetic defect can be actually collected. And then, of course, for the genetic defect, the fluid and the fetus have the same DNA. So if you can collect the amniotic fluids, then you can test for the DNA. So under local anesthesia, then a thin needle is inserted through the woman's abdomen and into the womb. Then about 20 milliliters of the blood can actually be withdrawn and then sent to the lab for evaluation. So test result often takes like 1 to 2 weeks. And from there, then you can check and see whether the child is having a chances of coming off with with the sickle cell. And then, of course, there is also, and the other one is cryonic villus sampling, and it involves the removal and testing for a very small sample of the placenta during any pregnancy. And the sample, which contains the same DNA as the fetus, is removed by catheter or a thin needle inserted to the cervix or by a fine needle inserted through the abdomen. So the tissue is tested for genetic changes identified in an affected family member. So these results are usually available within 2 weeks. So these are the two types of tests that you can do for the genetic counseling to know whether your baby is having a chances of coming up with the sickle cell disease. You can use cryonic villus sampling and then amnioinate, yes, and amniocentesis. So then, of course, what are the treatments? So the treatment is usually aimed at avoiding crisis, relieving symptoms, and preventing complications. So babies and children aged 2 and younger with sickle cell anemia should make frequent visits to doctors. And of course, children older than 2 and adult are expected to visit a doctor at least once a year. Then treatment might include medications to include pain and prevent complication as well as the transfusion. Then medication, antibiotics is always needed because to prevent the infections due to the damage caused by the sickle cell anemia, the spleen. So spleen, yeah. So usually a child of 2 months is expected to be taking penicillin. That is for those children that have sickle, so they are expected to be taking penicillin from 2 months to at least 5 years old. So during so or doing so helps prevent infections such as pneumonia, which can be life-threatening for an infant or child with sickle cell anemia. So as an adult, if you have had your spleen removed or had pneumonia, then you might need to take penicillin throughout your life. So then the medications. So pain relieving medication is also important to be relieving pain, and there is also need to take hydroxyurea. For example, we have Broxia, Hydratia. So when taken daily, hydroxyurea reduces the frequency of painful crisis and might reduce the need for blood transfusion and hospitalization. So then Another treatment, so assessing stroke using a special ultrasound machine that is transcranial, doctors can learn which children have a higher risk of stroke. So this fearless test, which uses sound waves to measure the blood flow, can be used to on children as young as 2 years. Then regular blood transfusion can decrease the stroke risks. Then vaccination to prevent infection. So children vaccinations are important for preventing disease in all children. So vaccinations such as pneumococcal vaccine and the annual flu shots are also important for adults with sickle cell anemia. So blood transfusion, you know that actually in the case of blood transfusion, in the red blood cell transfusion, red blood cells are removed from a supply of donated blood, then given intravenously to a patient with this class. So you know that in the blood we have red blood cells and also non-red blood cells. So you separate the red blood cells from the others and then you now deliver it to the sickle cell anemia patient. So red blood transfusion increases the number of normal red blood cells in circulation, helping to relieving anemia. So in children with a sickle cell anemia, higher risk of strokes, regular blood transfusion can decrease the risks. So, and then treatment. Still, nitric oxides. So, people with sickle cell anemia have low levels of nitric oxides in their blood. And of course, the major importance of this nitric oxide gas, it helps keep blood vessels open and reduce the stickiness of the red blood cell. So, treatment with inhaled nitric oxide might prevent sickle cells from clumping together. So study on nitric oxide have shown little benefit so far. Nitric oxide is very, very important in the treatment of anemia. Then bone marrow transplant. So we know that the red blood cells are usually produced in the bone marrow. So the bone marrow transplant is important. Yeah, so bone marrow transplant, also known as stem cell transplant, offers the only potential cure for sickle cell anemia. So, but it is usually reserved for people younger than age 16 because the risk increasing for the people older than 16. And sometimes, if you are older than 16 and you check sickle cell anemia, there is actually a serious risk associated with it, including that. So a bone marrow transplant involves replacing bone marrow affected by a sickle cell anemia with healthy bone marrow from a donor. But the procedure usually match or uses a matched donor. So it's not every bone marrow that you just say will transplant. No, you have to know there it must be matched. Maybe from a sibling, and you can't do that. So for many donors are not available, but you can use stem cells from the umbilical cord to generate the stem cells, the bone marrow, and then you now use it for the transplant. That is stem cell technology. And then bone marrow transplant. Okay, all these are related to the bone marrow transplant. So it's usually recommended to a child. Yeah. So then the next one is experimental treatment. So you can use gene therapy because we know that in sickle cell anemia, we said that the problems is actually started from the substitution of adenine with thymine, which leads to the production of non-polar amino acids, valine, which replace glutamic acids. So using aging therapy, you can hit a gene, a normal gene, and then you can now insert that normal gene into the bone marrow of the people with the sickle cell. Of course, this result in the formation of the normal hemoglobin. So scientists are also exploring the possibility of turning off the defective gene while reactivating another gene responsible for the production of the fetal hemoglobin. So a fetal hemoglobin is a type of hemoglobin found in newborns that prevents sickle cell from forming. So potential treatment using gene therapy are a long way off, however. So of course, in the experimental treatment, although several of the initial hurdles to sickle cell gene therapy appear to have been overcome, so it is a prudent to recognize barriers that remain. So we have gene augmentation therapy. So in gene augmentation therapy, you just get a normal gene cell with non-functional gene, then you get the functional gene, and then you remove the non-functional gene and then replace it with a normal one. So that is a gene augmentation therapy. So efficient transduction of the human sickle cells with the lentiviral vectors. So usually the gene delivery is we use a virus, a lentivirus vector to deliver the gene. But the complicated component of many globin vectors present unique challenges for the production of high titer virus capable of robust transduction. So experimental treatment, so scaling of result to multiple patients is a non-trivial challenges. So gene engineering method are rapidly evolving and should facilitate the development of second generation. So gene therapy approach is coming in the coming years after many years of preclinical laboratory investigations. So gene therapy options are now on the horizon for patients with sickle cell disease. So ladies and gentlemen, thank you for listening. And if this is the first time you are coming to my YouTube channel, please subscribe. Thank you.